Hey guys, welcome you back to my channel by Mixup. Hope you're doing well. So in this today's video, we are going to discuss about the regulation of gibberellic acid. So what is this gibberellic acid and why it is needed? Gibberellic acid, it is a phytohormone which is present in the plant, and this is needed for gaining the tallness of the plant. When an exogenous gibberellic acid has been applied to the plant, it attains its height. Okay, when in the mutated plant, this gibberellic acid is applied exogenously or from outside, it can gain its height. As well as when the experiment was done in the grapes, this grapes which was present, it was attaining the small size. So when there was application of the gibberellic acid, it gained its size into the last grapes was formed. Here you can see that the grapevine which is present here it is small and here it is large when the GA was applied in it. Okay, so the regulation of this gibberellic acid or other phytohormones which are present in the plant, it is basically through the degradation machinery. So who is degraded over here? So the repressor protein which are present, it is being degraded. Okay, so let us understand how this is working, how this degradation of this protein is working in case of gibberellic acid. So when in presence of gibberellic acid, this GA bind with a protein which is known as GID1 and it recruits the repressor which is the DELA protein. Okay, here after the recruitment of this repressor, it forms a complex and this complex is been polyubiquitinated. So this repressor protein is being polyubiquitinated. So for that it needs to form a complex with the SLY and SKP1. Okay, so this is the E3 ubiquitin which is present, and this E2 which is present over here, it carries a ubiquitin with it. This ubiquitin which is present, it is being transferred into this repressor protein. Okay, it is being transferred into the repressor protein, and after the polyubiquitination, that means formation of multiple U, attachment of multiple U in this site, it forms a polyubiquitination site. After which this target protein which is present, it is being degraded by 26S proteasomal pathway. In this way, the regulation or the degradation of protein is taking place in the gibberellic acid. So let us see how it is regulating other transcription factors or other proteins. Like you can see here, there is a repressor which is present, and there is a protein like PIF34 which is binding with this promoter region of gibberellic acid, promoter region of this DNA, who is regulating the formation of the gibberellic acid related genes. And in absence of gibberellic acid, this is in absence of gibberellic acid. In absence of gibberellic acid, the repressor, it is binding with this PIF34. Okay, due to this PIF34, it cannot activate the gibberellin regulated genes. So due to this repressor, there is a inhibition of this gibberellin regulated genes. In presence of gibberellic acid, when this gibberellic acid is present, or when we keep the gibberellic acid from exogenous or from outside, it forms this gibberellic acid, it binds with GID1 and it recruits the repressor protein. Okay, it recruits the repressor protein, and this repressor protein is having a portion which can bind with the SLY1 complex, and the E3 ubiquitin ligase complex is binded over here. Now here the E2 is carrying the ubiquitin in it. It is carrying the ubiquitin in it which is transferred to the repressor protein. After which there is formation of poly U over here. There is poly U over here, and there is degradation of this repressor by 26S proteasomal pathway. So here the repressor is being degraded. So as the repressor has been degraded, now this PIF34 is now free. It can bind with this promoter region of this DNA, and it help in the transcription of the gibberellin related genes, and there is gene expression. Due to which the work which was done by the gibberellic acid, it can do its work. Now no one is inhibiting it. So in presence of gibberellic acid, the repressor is being degraded and there is formation or there is synthesis of the gibberellin related genes. Okay, so now let us understand why is the requirement of the gibberellic acid needed in the plant. So what happens, this is the embryo which is present. Here the gibberellic acid are been synthesized in the embryo. In the first it has been written that gibberellins are synthesized by the embryo and released into the starchy endosperm by the scutula. So this is the embryo where the gibberellic acid has been synthesized and it is been transferred here into the endosperm. Okay, so it has been transferred into the endosperm through scutellum. Okay, now next, now this gibberellic acid which is present in the endosperm, it is being transferred or it is being diffused in the aleuron layer. Okay, and here it is being transferred into this aleuron layer. Now next is the aleuron layer are induced to synthesize and secrete alpha amylase and other hydrolases into the endosperm. So what happens in the aleuron layer, there is formation or secretion of alpha amylase and other hydrolase. So these are the enzymes which are synthesized and they are again transferred into the endosperm. So these hydrolytic enzymes which are present, they are being transferred into the endosperm. Okay, now the starch and other macromolecules are broken down to small molecules. Here what happens, the starch as well as other macromolecules are being broken down and they are being transferred into this endosperm again. The endosperm solutes are absorbed by the scutellum and transferred to the growing embryo. So here the endosperms which has, so here what happens, the starch are been broken into small molecules, so this can be easily absorbed. So it is being absorbed and it has been transferred into the into this region, into the embryo through the help of the scutellum, through by bypassing the scutellum, and it has been transferred to the growing embryo. Okay, so now we have to understand how this is being synthesized, how this alpha amylose is being synthesized due to the presence of the gibberellic acid. So we will understand by this signaling pathway how this is being synthesized, how the alpha amylase is being synthesized. So there are different type of gibberellic acid like GA1, GA2, GA3, GA4. So this work is being done by GA1. The secretion of alpha amylase is done by GA1 because this is one of the active gibberellic acid which is present. Now we can see that the GA1 which is present, it comes from the embryo. It comes from the embryo and it enters into the aleuron layer. This is the aleuron layer. Okay, this is the aleuron layer. So it comes from the embryo and it enters into the aleuron layer. Okay, next, calcium dependent signaling transduction pathway involves calmodulin and protein kinase. So due to the presence of the gibberellic acid, it initiates a calcium calmodulin dependent pathway which is necessary for alpha amylase secretion. This is one of the work. Now next, what does this GA1 does? It binds with the GID1 and it forms a complex with the stellar repressor. Okay, now this ubiquitination, after it is binding with the repressor, after the gibberellic acid and the GID1 is binding with this repressor, what it does, it help in the polyubiquitination. So this DELA protein which is a repressor, it is having one domain which is known as the GRAS domain. This one is the GRAS domain. So the polyubiquitination is taking place in this GRAS domain. Okay, and there is degradation of the DELA protein, DELA complex. Okay, now this what this DELA complex was doing, it was repressing this. So there was no secretion of GAMYB gene. So now this repressor is being degraded. So here there is no one to repress this protein. So here there is secretion of this gene. Which gene is was secreted? That is GAMYB gene. Now this gene is being secreted over here. So here the mRNA is formed by the process of transcription. and this is being transferred to the cytoplasm for the formation of protein. This GAMYB, which is a transcription factor, it has got some proteins and it needs to be transferred again to the nucleus because it is a transcription factor. It is required for the secretion of some other gene. Okay, so this GAMYB, it comes and bind in the GRAS domain. Okay, this GRAS domain is, after binding with this GRAS domain, it helps in the secretion of the alpha amylase gene, or the alpha amylase gene is being transcribed in this region. Okay, after the transcription, it needs to be transferred for the formation of protein. So here the alpha amylase mRNA is being through the process of transcription, alpha amylase mRNA is being formed here. Now next it comes into the ribosome for the formation of protein. After the formation of the protein, it comes into this ER over here, and it is being transferred through formation of this vesicle. Okay, this alpha amylase proteins which are present, now it is after some of the modifications. Here there is some of the modifications in the endoplasmic reticulum, in the Golgi there are modifications, and they are pinched out from this part, and there is release of this alpha amylase. Okay, so what is this requirement? What was the requirement of this alpha amylase? They were required for the degradation of starch in the endosperm. Okay, already we have discussed here, they are required for the degradation of starch in the endosperm. In this way, the presence of gibberellic acid is helping in the secretion of the alpha amylase in the plants or in the embryo. This was all how this GA was helping in the secretion of alpha amylase. Now let us understand how the phytochrome and the gibberellic acid regulates the growth of this plant. So what happens in presence of light, the PR form is converted into PFR form of the phytochrome, and in this PFR form the phytochrome has been transferred inside the nucleus, and this PHY in the PFR form, it binds with the PIF, and now it is being ubiquitination, and now it is being degraded. So now there is no transcription of this PIF regulated genes. यहाँ पे जो PIF regulated genes का formation हो रहा था, यहाँ पे PIF gene का formation नहीं है, PIF regulated genes का formation नहीं होगा. अगर यहाँ पे if the PIF induced gene are not expressed here, the stem का जो growth है, it is being inhibited. ये जो stem का growth है, it is being inhibited. Now let us see what happens in case of low light. Okay, in case of low light, the PFR form is not present. The phytochrome which is present, it is in PR form. So it cannot bind with the PIF. So it cannot form a complex with PIF. So now this PIF which is present, it is not being degraded. Okay, here there is a protein, there is presence of gibberellic, and when the gibberellic acid is present, it binds a GID1 and it forms a E3 ubiquitin ligase, and it degrades the DELA protein. यहाँ पे DELA protein का degradation हो जाएगा. So here there is no repressor protein, that means no DELA is been degraded, so there is no repressor protein for the transcription, as well as PIF is present, that means phytochrome inducing factor is present over here. So here it can bind in this promoter region, and it will help in the formation of this PIF inducing factor. Phytochrome in that means the PIF means the phytochrome inducing factor, due to which there will be the growth of the plant. So there will be a promotion of the stem growth. This is how the gibberellic acid as well as the phytochrome is regulating the growth of this plant. Thank you everyone for watching this video till the end, and if you like this video, please do not forget to share, like and subscribe. Thank you.